right, so we are going to look at a special little area of the GI tract called the duodenum, the first portion of the small intestines, and the three really important accessory organs that are going to be found there: the pancreas, the liver, and the gallbladder. So just to refresh, this is a picture of the general idea of the digestive system. And what we're going to look at is kind of hidden behind the liver, but there's a special little area here that has a huge impact on our digestive system and our ability to break food down, chemically digest food, that is. All right, so this is a picture showing the whole GI tract, the alimentary canal stretched out, and I'm gonna focus in on this little area. Now, on this, you don't see the liver, but the gallbladder here, and again, sorry, since I enlarged this, it got really bad, fuzzy, but the gallbladder here, this green object, is there to store bile, and the bile is going to be produced by the liver. But you're going to have the liver, the gallbladder, and the pancreas all releasing special enzymes and chemicals into the chyme as it leaves the stomach and enters into the duodenum. And this is going to help break the food down. So this is a better picture of what it looks like, and we'll talk about this a little bit more. But you've looked at this in lab. In lab, basically, you're going to start here. You're going to have the common hepatic duct, which is leaving the liver. And then you're going to have the gallbladder, which is going to connect to that by way of the cystic duct. All right, so the cystic duct is here. And as those two join, the common bile duct and the cystic duct, this that's coming down is now called the bile duct, because that's all it's going to handle is bile. As it comes down, it will merge with this duct that's coming through the pancreas, called the pancreatic duct. All right. Again, that's kind of a review of some of the stuff you had at lab, but we're going to look at some special things also in here. There's an area in here that's a little bit larger where these two ducts join, and it is called, as it says here, the hepatopancreatic ampulla, down here at the bottom, the hepatopancreatic ampulla. And again, if you remember, ampulla just means larger than normal duct, and it's going to be controlled by the hepatopancreatic sphincter, which is going to allow blockage, and then when it's ready to release the substance, it will dilate and release the substance into the chyme. Now, to start with, we're going to look at the pancreas. The pancreas is one of the most amazing organs. It's got both major endocrine and exocrine properties. Now, the endocrine is going to be about hormones. It's going to be insulin and glucagon. We will look at those in the endocrine chapter. Right now, I'm not worried about these. Right now, since we're in the digestive chapter, we're looking at the exocrine portion because it is part of the digestive system. Now remember, exocrine portions have ducts, and that's what we just looked at. The pancreatic duct, that is going to be associated with the exocrine portion of that gland. As it produces the digestive enzymes and fluid, the fluid itself, as a general rule, is called pancreatic juice. That is what is going to be releasing into the chyme. Now, again, in lab, you looked at the fact that the pancreas has special little areas. Now, you're going to have things called pancreatic isle cells, which are going to be associated with the endocrine system. But the other cells, the majority of cells, are called acinar cells. And these are the ones that make up the majority of the pancreas, and they are the ones associated with the exocrine system. They make the pancreatic juice. Now, there is a pancreatic duct which goes along and collects it. And it's going to empty that pancreatic juice into the duodenum. Now, the pancreatic duct and the common bile duct join to form this very short but dilated, which just simply means larger than normal, tube called the Hepatic ampulla. Now remember, hepato means liver, which both all of the bile is going to be coming from the liver. The gallbladder just is storing it. Pancreatic, saying that that's the other part joining it. And then ampulla simply means larger than normal duct. And it is going to be also controlled by a well-named sphincter called the hepatopancreatic sphincter. You do not have to worry about it being the ampulla of Vader or the sphincter of Odai. That sounds like something off of a Marvel Universe thing. But anyway, those are the parts of the pancreas that you're going to need to know. This again just shows a little picture of it. It's a little bit more enlarged. I'm not really going to spend much time on this. Now the pancreatic juice, a lot of people get very frustrated with having to learn enzymes, but again, if you just get the basics of it, you can usually work things out pretty well. Remember that enzymes are generally going to end in this ASE, and it will tell you what they work on. Amylase works on amylose, and amylose is a carbohydrate, a complex carbohydrate that plants make. And so when it says pancreatic, which is telling you where it's coming from the pancreas, amylase, it is going to break down amylose, which is a starch. Now understand, starch and glycogen are both polysaccharides, which means they are long chains of individual carbohydrates or monosaccharides. As this pancreatic amylase starts to split these large molecules down, they break them into what we call disaccharides, which are simply two simple sugars. All right, so it still needs to be broken down into the monosaccharides, but at least this makes a good start to getting it taken care of. The next one is going to be this pancreatic lipase. Again, pancreatic's telling you it's coming from the pancreas. If I cut this here, this lipase, ASE, tells you it's an enzyme, and it's going to work on something called lipids. Again, hopefully everyone knows that lipids are fats. And the main dietary lipid we looked at is something called triglycerides. Remember, a triglyceride is going to have a glycerol backbone and 3 fatty acid chains. So it's going to try and break this little bond here. Now, the next several are going to be things dealing with proteins. Now remember, proteins are long chains of amino acids. And these amino acids are held together by peptide bonds. And these peptide bonds, these long chains of amino acids, are going to be curled and coiled and all twisted up to form a unique structure. That's what a protein is. You can have a polypeptide, which just means a really long chain of amino acids, but if it's not twisted and curled, it is not a protein. Now pH, one of the very first things that pH does is it takes those curled up, twisted up proteins and unravels them into chains of polypeptides. And that's how we start beginning to break it down. All right. Now again, remember, they can be thousands of amino acids long, so they can be really big proteins. Now the trypsin, the chymotrypsin, the carboxyl peptidase, all 3 of these are designed to break down proteins. Now, this trypsin and chymotrypsin, these are both going to be stored inactive, and they're going to be there's going to be a chain of events that cause them to interact to become active. All right, so trypsin is released as trypsinogen. Remember, OGN means it's an inactive form. When it gets active, it will help digest proteins, but its big thing is it is going to go in and activate chymotrypsin. As you can see, chymotrypsin is activated by trypsin. Now those two work together. Hopefully you can kind of see, since both of them have trypsin in the name, they are kind of held, they are kind of working side by side. I'll try and erase some of my scribbles here. Now, carboxypeptidase. Peptide goes into peptide bonds. This again, all of these you'll see are released as inactive because anything that's going to break down a protein, if it's stored active, is going to eat through the container. So you can also see that this is going to be activated by trypsin. Now again, on this upcoming test, it's not going to be super crazy on this. I'm going to have to use the questions that Connect use. I can go through and pick them out. What I want you to know really is before you get anything, first and foremost, these three are for proteins. Understand that these two are both activated by this one, and that's kind of basically it on that. All right. Now, the next one, so we've looked at digestive enzymes for carbohydrates, for lipids, and then 3 of them, it's hard to make a little check mark on this, 3 of them for proteins. So now we have, there's only one other major organic compound, and it is called nucleic acid. So we also have an enzyme that helps break down nucleic acids, and they are called nucleases. This is, again, this is, an enzyme, and you can kind of see ASE, it ends in ASE, so that should help you understand that it is an enzyme, and it's designed to break down nucleic acids. Now, think about this. One of the main nucleic acids is DNA, deoxyribonucleic acid. And so if you are eating steak, which is a muscle, it is going to have skeletal muscle has multiple nuclei, which means there's a lot of nucleic acids, and so this is helping break those down. And then finally, one that is not dealing with dealing with the organic compounds is bicarbonate ions. We talked about this several times. Bicarbonate is a buffer, so this is going to help. Because again, the pancreas is going to be associated with the duodenum, the first part of the small intestines. The chyme that is leaving the stomach is going to have a very low pH, which is another way to say it's going to be very acidic. So right away, the duodenum is going to send a signal and have the pancreas release bicarbonate ions to try and buffer the pH of that chyme that's coming in. Now, there's 2 major hormones that help regulate the release of the pancreatic juice, and the first one is called secretin. I'm starting with secretin because secretin is the one associated with bicarbonate ions, the last thing we looked at. So as soon as the chyme, this acidic chyme, enters the duodenum, it's going to cause release of this substance called secretin, as it shows here. And secretin is going to go back, and it's going to go to the pancreas and cause a release of bicarbonate ions. And this is going to help neutralize that chyme. The other one is one that is in everything, it seems. This thing, cholecystokinin, or CCK, this is going to stimulate the pancreas to secrete the pancreatic juice. So as this chyme comes in, CCK is going to be released. It's going to cause the pancreas to secrete the enzymes that were in it into the chyme. But remember, the other thing that it does is it's going to go back to the stomach. So when it's released, not only does it go back to here, but it's going to go back to the stomach up here and cause the stomach to decrease its motility. It's basically telling it to slow down, let the duodenum handle what it's been given. So from that point, we're going to look at the liver. Now, the liver is, and you do need to know this, it is the largest internal organ, right? Now, the largest organ, remember, is the cutaneous membrane, which we call the skin. But the largest internal organ is the liver. It is located in the upper right quadrant of the abdominal cavity. And it is obviously well supplied with blood vessels. That should kind of be and I can't write with this, but that should actually kind of say duh after that, because that is something that should be kind of common sense. But anyway, the liver is what we're going to talk about right now. Now, the liver, you do need to know that it has 4 lobes. I'm not worried about the different ones, but know that it has 4 lobes. That's not going to be a big part of it. I can't tell you, it won't be a question on it. I'm definitely not going to ask you the names of the lobes, but you should know it has lobes, 4 lobes. But the big thing is, inside there are these lobules, these little 6-sided sections called lobules, and they have a very interesting makeup. Now, we have to understand that the liver, I'll try to draw something up here, that's supposed to be the liver. I am going to have a blood supply coming to the liver that's going to be carrying oxygen, right? And then I've got my my intestinal tract, which I'll just kind of draw like that. And the intestinal tract, I'm going to have some blood coming into the intestinal tract that's going to be picking up nutrients. It's going to be dropping off oxygen to those cells, but it's picking up nutrients, and then it's going to go back to the liver. So this is going to have a low oxygen content. That's supposed to be that, but high in nutrients. And I'm not going to try to draw that. So both of these are incoming blood supply. This one here is called the hepatic artery. This one here is called the hepatic portal vein. Now, as they mix, then I'm going to have another vein going back out to the heart. Getting better at this a little bit. That is called this one is called the hepatic vein. All right, so we have this one here called the hepatic artery. We have this one here called the hepatic portal vein, and then this one here called the hepatic vein. All right, so hopefully you get those. Now, what I'm going to tell you is that I'll try to selectively erase some of this stuff so that we can see it a little better. These two here, the 2 incoming ones, are what we're going to look at. This is the hepatic portal vein. You can see it's blue because it's deoxygenated. It's the one that's coming from the small intestines. And then this is the hepatic artery. This is the one that we looked at that's coming from the abdominal aorta. So they're both bringing blood in. Right? This one's dropping off oxygen, but what's going on with this hepatic portal vein is all of these little cells through here, these are little hepatocytes, or what they're called, which just means a liver cell. The hepatocytes are modifying everything we've eaten and trying to regulate what's going on. Now, one of the byproducts that's going to happen is, as they start seeing red blood cells, erythrocytes, that are older, they will start taking them apart, and part of the byproduct of that is something called bile, and bile is going to move the opposite way and go out by way of this bile duct. So I have 2 things coming in, and then this third that is exiting. Right? So this is actually called the triad. And we'll look at that, I believe, in the next slide. Yeah. So this slide shows the hepatic portal vein, the hepatic artery, and then on this part here, we've got the bile duct that's removing the bile. So again, as it goes through there, and you do need to know that liver cells are called hepatocytes, and as these hepatocytes work and modify all of the nutrients that are going through, as well as the blood, it is going to start taking down old red blood cells and modifying it, or just taking it down and recycling the liver as it can. But part of the byproduct is bile, which is going to be removed by the bile duct. Now, the liver has many, many, many important functions. It's, you have a problem with the liver, it's a bad deal. So the liver has a bunch of different things. I'm not going to worry too much about this on here. it's going to convert glucose into glycogen, and if we have extra glucose, if we have a deficiency in glucose, it's going to actually break down the glycogen into glucose. That's a cycle that we're constantly going through. It's this big cycle of how our body works, right? So we have extra glucose, we store it as glycogen. When we're deficient in glucose, we take the glycogen, break it down to release glucose. Things that are non-carbohydrates, if we have extra, it will also convert it to glucose. Again, not worried too much about some of these things. We'll talk about lipoproteins. If you've ever heard of HDL or LDL, that's what it is. It is a lipid and a protein. These are basically carrier molecules that help lipids or fats flow through our body without accumulating and causing problems. Phospholipids, these are main things in cell membranes, and cholesterol, which is all lipid hormones, have their base in it. Now again, I'm going to kind of skip through the rest of this. I do want you to know down here it acts as a blood reservoir. This is a pretty big thing that I can see that being a question. Understand the liver is a big area can be used as a blood reservoir, so to speak. Again, this is kind of a basic idea. I'll tell you, it's probably a pretty good slide to know where it is as you go farther along, especially in the healthcare field. A lot of issues come up with the liver, and so this is kind of a decent thing to know where it is. I plan to try and take these out, but I did want to point out one thing that I think you should know, that the liver is capable of regeneration if it has at least 30% of it being healthy. And so that's kind of a pretty cool thing, but I just wanted to point that out. Now bile, the thing about bile, understand that it is yellowish green. All right, it is constantly being produced by the hepatocytes, and if you have a gallbladder, you can store it. If you don't, it just backs up into that bile, the common bile duct, and releases itself slowly into the small intestines. But it doesn't. It really can cause a problem with your ability to break down lipids. Now, the components of bile are, water's the main one. You got bile salts, which is this is the one we're really concerned about in the digestive system, because it's going to be about breaking down fats. The other thing is it's going to have bilirubin and biliverdin. We talked about this and how all bilirubin and biliverdin is the breakdown and unraveling of hemoglobin. So if a lot of people have heard of bilirubin. If someone has bilirubin in there will end up being very yellowish. It's a big thing with babies because their liver isn't functioning. A lot of times babies' livers isn't functioning at 100%. So there's a spillover of bilirubin into the bloodstream, which is not a life-threatening disease or whatever. And they usually put them under a UV light and it breaks it down. But if you've ever had a bruise that was so bad it went from black and blue to green and then finally yellow, that's what those are. And that's why the bile is a greenish yellow liquid. So this one really, however, is the main one we're worried about, emulsifying fats. And we talked about what this is. I showed you a little video. I'll probably try to upload it in here, just kind of showing you what happens when you mix emulsification substances into a liquid. And I showed it with soap and then olive oil and water. I meant to take this slide out. Don't worry about it. But what I want to talk about now is this bile, which is stored in the gallbladder. All right, so as long as you have a gallbladder, the bile is constantly being produced, from the liver, and then it comes back here and it gets stored in this big green pouch called the gallbladder. Now, remember that the gallbladder joins the common hepatic duct by way of the cystic duct, and after that, from that point on, it is referred to as the common bile duct. It empties into the duodenum via the hepatopancreatic ampulla. All right, so all those we've already talked about. Now, the gallbladder is going to generally concentrate those bile salts and bile pigments. Sometimes cholesterol can fall out of solution, which that precipitates, which just means it clumps together basically, and it will form these solid crystals, and they call them gallstones. Excess of bile concentration, too much cholesterol secretion by the liver, and inflammation of the gallbladder. It can be a really serious problem. This is why a lot of people will get their gallbladder removed. Now, we're going to talk about our friend CCK, cholecystokinin, because not only does it go back, and cholecystokinin, remember, goes to the to the pancreas, causes it to release its substance. It goes to the stomach and causes the stomach to slow down. But it's also going to go back and go to the gallbladder, cause it to squeeze and push the bile out. So it's going to release, it's going to cause the bile to be released into the duodenum. And so cholecystokinin has three different things. It works on both the stomach, it works on the pancreas, as well as the gallbladder. So this is kind of a little breakdown of the different hormones in here. Of the digestive tract. I would kind of read over that. I mean, again, not going to be real specific about a lot of stuff. I'm not worried about you knowing the parietal cells of these. The only ones, there's two that I want you to know the cells. One of is pepsin or pepsinogen, which is chief cells. The other one is hydrochloric acid, which is the parietal cells. Outside of that, I'm not worried about the different cells that secrete them. Now this goes back to the bile salts and emulsification. Again, we looked at having a video. I had that video that showed how emulsification happened, where I put the olive oil in the water and I dropped some soap in it and stirred it up. I might put a video together that kind of shows that, but what that does is it increases the surface area of those, because instead of it glumping all up, when it has this emulsification, it breaks it down into lots of little droplets, and this is going to increase the surface area of the lipids. I'm going to erase all of this. Wow. And so hopefully that makes sense, because what's happening is our body is mainly water. And water, in the especially the small intestines, the chyme is mainly water, because water is what drives all chemical reactions. So if we've got a lot of lipids, they want to pull together because they don't deal with water. Oil and water doesn't mix. So emulsification helps it mix, and this is what enhances absorption of the fatty acids and cholesterol. It says forming micelles, which is just a basic way of saying small droplets, or scientific way of saying small droplets. And anything that's fat has to have this to really be absorbed. So we're going to look at, there's 4 fat-soluble vitamins. You will have to know them: A, D, E, and K. Those are all fat-soluble vitamins. And so this bile is needed to help absorb those. And almost all bile salts are recycled.